everyone, today I'm going to go over microbiology lab tips. So one thing that's going to be done a lot in microbiology, a lot of microbiologists do this, is a thing called serial dilution. And what this is you're taking essentially a broth culture from a stock solution and you're diluting it down until you can get a easily countable and measurable amount of bacterial colonies on a smear plate. So in order to do this, you'll take 1 milliliter of the original culture, the stock culture, and then you're going to dilute it in 9 milliliters of solution in separate test tubes. And what this does is it gives you 1 milliliter of solution in total. And this kind of helps keep your methods consistent, so that way you can easily do calculations. And you accurately know what your dilution is. You'll know it's 1/10, 1/100th, 1/1000th, so on and so forth. And so what you're going to want to do is you're going to plate 1 milliliter of the solution onto the bacterial plates. And then you're going to let it grow. And then the colonies, you're going to look for the colonies that you can count. So here, as you can see, there's too many to count. So this would be a bad candidate for serial dilution. 500 is still pretty bad. And I believe 6 is way too low. So you're going to want to take this one for 71. And so the formula you're going to use to count the bacterial colonies is the number of colonies growing on the plate times 1 divided by the volume used to put bacteria on the plate times 1 over the dilution. So here's an example. When 1 milliliter of broth culture is plated from a tube diluted 1/100th and the plate grew 230 colonies, the formula would be 230 times 1 over 1 ml times 1 over this number right here, which would give you this. Most of the time you're going to want to put it in a scientific notation, 2.3 times 10 to the 4th. Okay, here's another practice example to work. So let's, from our dilution, we took 1 milliliter of the stock solution, and then we diluted it, and then we put it onto our plates. And like I said here, there's too many colonies. And here is a thing called too few colonies. So you want to pick the 159. Okay, so we chose 159 times 1 over 1 ml times 1 over 1000. And the reason why it's like that is because of this. And this is why it's done essentially with 1 milliliter, because it keeps our numbers consistent and we can easily do these calculations. Okay, so the next important thing that's going to happen is gram staining. And this is probably one of the most widely used and very important subjects to know. You have two types of bacteria, the gram-positive bacteria and the gram-negative bacteria. Here are the gram-positive. They are purple. The gram-negative bacteria are a pink or a red. They, depending on whatever dye you use, they may come out pink or they may come out red. Okay. So the procedure to do gram staining. You're going to prepare your bacterial smear. You use 1 drop of inoculum in water. You got to heat fix the bacteria smear. This is very critical because it not only kills your bacteria, it also fixes it to the slide. So that way when you stain crystal violet and then do the water wash, you don't wash away your bacteria. Okay, so the next step, stain crystal violet, and then you're going to wash with water. And then you're going to treat with iodine. And now what happens is, once you treat with iodine, if it's gram-positive, it's going to stay purple. However, this is where it's going to differentiate between gram-positive and gram-negative. The iodine, see? Once you move on to treat with alcohol and the decolorization phase, the gram-negative is going to lose that purple color. And then you're going to counterstain with saffron. Wash with water, blot dry, and then you'll know if it's gram-positive or gram-negative, because gram-positive will be purple, gram-negative will be pink, because of the treatment with the decolorizer and the iodine. That's very important right there. Okay, so what does all of this mean? It's all well and good to just know how to stain it, but what does it mean? Well, gram-positive stains purple. What that means is it has a thick peptidoglycan wall. So, purple positive peptidoglycan. I don't know if that might help. Gram-negative stains pink, means there's thin to no peptidoglycan. So, gram-negative, no peptidoglycan. So think about it kind of like that. Gram-positive has the 3 P's. Positive, purple, peptidoglycan. Gram-negative, you're looking for the double N. Negative, no peptidoglycan. So hopefully that helps you guys remember. And there are other types of staining processes, but they are less so used. You have the endospore staining, and but they're less widely used, and the way you do it is essentially the same. And the only difference really is that you use different colored dyes. And I hope this helped you with a quick overview of laboratory techniques. Have a great day.